Good morning and welcome to Peptidream's R&D Day presentation. This is Yan-Ting Chen, Head of Business Development at Peptidream. With me today on this call is Patrick Reid, Chief Executive Officer of Peptidream. During today's call, Patrick will provide some recent updates and perspectives. At this time, all participants are in a listen-only mode. After the presentation, there will be a question-and-answer session. I would like to remind you that this call will contain remarks containing Peptidream's future expectations, plans, and prospects. Actual results may differ materially from those indicated by these forward-looking statements as a result of various important factors. In addition, any forward-looking statements represent our views only as of the date of this call and should not be relied upon as representing our views as of any subsequent date. We specifically disclaim any obligations to update such statements. Today's call is recorded and will be disclosed on the Peptidream website. And with that, I'd like to turn the call over to Patrick.
Good morning, everybody. Thank you for attending our 2022 R&D Day. I could go to the next slide, eh? Slide, eh? Today's agenda is we're going to run through just a brief introduction of the company and overall strategy, and then we will give some recent highlights across our peptide and small molecule programs, our PDC programs, a kind of newer area for us in the MPC space, and then of course highlight some of the recent updates around the radiopharmaceutical business and programs, and also talk about the growth factor business. And then we'll follow that with a Q&A session where we're happy to take any questions from any of the participants. So as everyone knows, we basically operate across two business segments, the drug discovery and development segment, and also now the radiopharmaceuticals business segment. And this encompasses four business strategies, three of which are the collaboration drug discovery, the PDPS technology licensing, and the strategic alliance or in-house partnerships and or programs. And then in the radiopharmaceutical business segment, of course, is the R&D manufacturing sales of radiopharmaceuticals and diagnostics. Both of these business strategies, or sorry, all of these business strategies have various key points to them and key contributors to the success of Peptidream. As everyone may be aware, Peptidream has continued to grow year over year from our inception in 2006 to 2022 here, we're now roughly around 200 plus scientists at Peptidream or staff at Peptidream. And now with the acquisition of PDR Radio Pharma business, we are in total now, the combined business is around almost 700 employees. In addition, on the left side of this slide, we've continuously grown our R&D capabilities, evolving from a company in 2010 that really was focused on just hit identification to now a company in 2022 that can do the entirety of the preclinical discovery and development process. And of course, as everyone knows, now with PDR Pharma, in the case of RI peptide programs, we can take these all the way through commercialization and marketing in Japan. And so these increase or expansion of R&D capabilities are key contributors to greater roles in partnerships, more controls over the programs that we actually progress, certainly larger deal financials. They're contributing significantly to larger deal financials, and also the ability to take internal programs further before we actually seek out-licensing and or partnering opportunities. So as we've discussed often, PDPS is still at the core of everything we do at Peptidream. This is a very powerful peptide discovery platform. And I won't walk through every aspect of this slide, but there's been continuous improvements to this platform year over year. And just on the upper left, we've expanded the amino acid building blocks that we use to create these libraries from many years ago having around 200 or 300 to having more than 3,000 nowadays, which makes us by far the leading company, global leading company in this type of area and space. These libraries allow us to make trillions of peptides in a single library. And now that we've joined this in the last couple of years with automation, with an automated platform, we can run a large number of library selections in parallel at a very high rapid speed. And this drastically, one, improves the amount of time that we can do things, and also continues to contribute to higher rates of of hit finding success. So, and this entire technology, sorry, just stay on that slide for a second. Hold on there. The technology, of course, is continually covered by a broad array of patents, to which we continue to file on new improvements to our core technology. One other aspect I just want to briefly highlight before we get into the kind of the therapeutic programs is the PDPS technology transfer or licensing business strategy. As many of you are aware, we have licensed out the PDPS technology to now 11, non-exclusively to 11 companies. And these licensees are actually working on a number of programs. And it's important to note that these programs are not included in our disclosed program numbers. And will not be included until they actually reach phase 1 or enter the clinic. And so those deals are structured in a way where we're eligible for development milestones starting from phase 1, and of course then royalties on any products that actually reach approval in market. In 2022, we announced this exclusive licensing deal with Fujiro Bio, which is the largest in vitro diagnostic player in Japan. And so we were quite excited to be able to announce this deal in September of this year. The peptides that are identified, our macrocyclic peptide seeds that are identified from our PDPS technology, we utilize those starting points to turn those into peptide drugs, which is largely where we started as a business. And then we also evolved to be able to turn these seeds into, in certain cases, to turn these into small molecule drugs, of which we have an example of that today. And those two modalities, both peptide drugs and small molecule drugs, have a number of unique advantages to them, which continues to contribute to the exciting progress of a variety of programs. And as we've mentioned in the last couple of years, oral administration of peptide drugs has become a more robust route and a more possible route than in the past, and it's contributing to a number of exciting advancements, we also believe. In addition to that, using these peptide seeds to turn into PDCs. So we've discussed PDCs probably for the last 4 or 5 years. We've already have a number of deals in this space, and utilizing these peptides to deliver a radioisotope, so these peptide RI conjugates, or to utilize these peptides to deliver an oligonucleotide payload, in the case of these peptide oligo conjugates, and then also peptide to cytotoxic conjugates, which is more similar to the classic ADC kind of concept. And we'll talk about each 3 of these areas, which have been our initial main areas of focus as far as payload modalities. In addition to that, there's a 4th section now that we're starting to talk about more, and will be from now on, around multifunctional peptide conjugates. And this is actually linking 2 or more peptides together to make bifunctional and trifunctional therapeutic molecules. And so we'll briefly touch on this area also, to which we actually have, of course, some examples. So just starting on this slide 8, at least in the top 12 programs across our pipeline. And again, there's an additional 26 programs not listed in this slide. They're in the lead to GLP or GLP to IND stages. But today we will highlight the 5 programs highlighted here in yellow. So as everyone is aware, our partner program with BMS is around a next generation PDL1 inhibitor. The development status of this program, it entered a phase 1 trial in the UK in April of 2022. And the study design is, of course, testing the safety and tolerability and the pharmacokinetics in healthy volunteers. It is being run through a CRO, of course, out of the UK, and we've listed the identifying number for this. The potential benefits of this next generation inhibitor, certainly it has different PK ADME properties from the existing or approved antibody therapeutics. In addition, I think it has some other interesting attributes to this program that make it quite exciting. The details of this trial, BMS applied for the ability to not have to disclose the details of the trial because of the high commercial sensitivity of this program. And so as is listed in the bottom there, the trial results do not need to be made public until 30 months after the end of the trial, according to the disclosure rules. And so this is a program, again, because of its high commercial sensitivity, that we will have to wait until we're able to make more public statements in regards to the progress of this program. But it represents a very exciting program for us, and we look forward to being able to announce as soon as possible. A second program in the pipeline is our GHR antagonist, AZP3813, which we licensed this program to Amolyt. And this program is making very exciting progress. As we list on the left, AZP3813 is basically an alternative to Sumivert, which is a currently commercialized therapeutic from Pfizer for the treatment of acromegaly. And we've seen that this peptide replacement is significantly improved. It shows Sumivert is actually a protein drug, just for everyone's knowledge, shows a much more rapid and sustained effect and has a faster off effect than Sumivert has. And so it seems to have better efficacy and also better safety profile and is extremely promising. And Amolyt is actually currently IND enabling studies with planning to enter phase 1 actually sometime in the first half of 2023. Probably around the middle of 2023 is probably most likely. And they have been presenting a number of data at a number of conferences. This is data recently from their the Endo 2022 conference. And they're actually slated to be presenting data in a number of conferences in 2023 on this exciting product and program. And we're able to see, just for everyone's information, normally in the upper right, it's usually easiest to see pictures, but the treated animals, which normally get very large very quickly, shown with the vehicle, when they're treated with this compound, they grow much more normal and don't show the, say, negative attributes of the acromegaly, which is basically a growth hormone-derived disease. And so this compound shows a very nice suppression of these IGF signalings, which is extremely important to this disease, in a dose-dependent manner, and actually can maintain these IGF levels for a long period of time, which is quite critical to the actual efficacy of this. And so we're very much looking forward, and you can see the results here, that we actually see very nice suppression in influence on the growth rate, body length, and anatomical parameters of this compound. So both ourselves and Amolyt are very excited about the progress and future of this candidate. Another program is our Peptide, is our SARS-CoV-2 spike protein S2 inhibitor, PA001. Shown on this slide, of course, is some of the in vivo efficacy data. When we've treated animal models with this compound, we can see a complete recovery in their body weight, and it's comparable to other existing antibody treatments that also target the spike protein. We can additionally have been able to observe that we can see decreased levels of cytokine release, which is largely driving a lot of the lung inflammation and lung challenges associated with these type of patients. And so on the right side is just showing we actually treatment with our compound ameliorates basically the lung inflammation that you classically see upon COVID infection. So we believe these very clearly kind of show the mechanism of action of this compound and its potential promise going forward. In addition, if you go to the next slide, Nick. This compound has a very broad potency across all of the strains of COVID that we've seen to date, which has actually not been common for any of the other therapeutic molecules out there. And so that also is a very promising attribute of this compound. This went into, as we had mentioned, and we announced earlier in the year, had gone through a clinical safety research study where we did a single dose escalation in healthy human volunteers. And the results returned, as is shown in this slide, really with a completely clean safety profile and no significant adverse events and or side effects seen. In addition to that, of course, we could observe a very nice, clear dose response PK profile for this compound. It represents potentially a first-in-class compound, certainly, with an extremely best-in-class safety profile compared to the other currently approved compounds for coronavirus. With that, of course, though, we'll talk in a later slide. we're still kind of considering the positioning of this compound as we look toward whether we would out-partner this program or whether we would consider taking this program into the clinic going forward. As everyone is well aware, the environment globally of COVID-19 continues to change, and so this is kind of an evolving study situation. The last program in this section is a therapeutic small molecule program that we have in partnership with Modulus, and so this is a program that initially started with a macrocyclic peptide, leading to the identification of a selective KIT inhibitor, which then led us to the ability to find a small molecule compound that functioned in a similar way. So this is the peptide being to enable the ability to find a small molecule compound. And this is a very, as is shown in the lower left panel, the other compounds on the market are not selective, do not have a high degree of selectivity for KIT, and is part gives some of their side effect and or risk profiles to those compounds. And this represents, we believe this compound represents one of the most selective KIT compounds that has ever been seen. And so this is very, very promising, which would mean it would have a much better safety profile and more tolerability. And they've seen, the Modulus side has seen very good data with this compound shown on the right of actually being able to suppress histamine release in these in vivo systemic anaphylaxis models, further confirming its potential use in allergic reaction. And so the preclinical proof of concept has been obtained in animal models for a variety of different allergic conditions. And the evaluation to identify the clinical candidate is actually ongoing. And out-licensing discussions are also progressing, and we are expecting news on this probably in the first quarter of next year, which we'll summarize at the end of this slide deck too. So next, we're going to move into just some highlights around the peptide drug conjugate programs that we have. At least in this top 12 programs that are described, 3 of these are related to PDC, 1 each with Reid, Novartis, and Takeda. And so we will briefly discuss the PDC space. So the ability to utilize our targeting peptides or our macrocyclic peptides to deliver a therapeutic payload specifically to a disease cell type and or tissue type is an extremely powerful use for our peptides in this platform. And the utilization of macrocyclic peptides versus antibodies really enables a greater array of drug payloads that can be conjugated compared to using an antibody. And as everyone is aware, we started initially on the left side of this, really focused on the delivery of radionuclides or radioisotopes for both diagnostic and therapeutic use. We've, of course, since evolved into also being able to use these oligonucleotide drugs, being ASOs or siRNA or even HDOs. And then we're also now evolving into cytotoxic or small molecule compounds as kind of our third class of payloads that we're investigating. And then there's some additional peripheral kind of aspects that are also we have been looking at as far as antibodies, conjugating other peptides, conjugating LNPs, some other exciting areas that potentially we will discuss more in the future as those advance. Just showing on the lower part of this, really these peptide carriers enable highly targeted delivery of various drug payloads to target cells in tissues. And we think they have multiple advantages as carriers compared to antibodies and will lead to the expansion, expanding the options for the drug payloads and discovery strategies. And so just highlighted in this table is the affinity and selectivity of our peptides is very comparable to ADCs. The size is certainly significantly smaller. Because they're chemically made, there's a lot more control as far as the ability to conjugate various payloads and actually purify those compounds to homogeneity, which can reduce certain aspects, risk factor aspects. And there's also differences in clearance pathways in PK and pharmacokinetics in comparison to the ADCs. So there's a number of differentiators that we think are could be quite powerful in expanding kind of the ability to use targeted therapies. And so initially just on the RI-PDC or the peptide radionuclide conjugates, as everyone is probably aware, a carrier that could deliver a high amount of this radioisotope payload directly to the cancer cell would allow for a much more targeted approach and also maximize basically the efficacy of that RI compound to be able to target the cancer specifically and have less, say, side effects on normal cells or normal tissues. And so we've listed some of the advantages of peptide RI conjugates on the right side here, some of these are key differentiators between being able to use antibodies, for example, for this approach. And so, such as the high selectivity, the PK properties of these compounds is very tunable. And in fact, the half-life of the peptides is much more in line with the half-life of the radioisotope payloads that we're actually conjugating. And that's in part why actually using antibodies to deliver RI isotopes has been quite challenging. And you're not seeing many programs actually advancing utilizing antibodies in this context. And in fact, again, there's currently two approved therapeutics on the market, targeted therapeutics on the market, Lutathera and now Pluvicto. And Lutathera is actually a macrocyclic peptide with a RI conjugate, and then Pluvicto basically looks like a small molecule or peptidomimetic with a small molecule. So certainly being on the smaller side seems to be very important or beneficial in comparison to using these large biologics for this type of targeting aspect. So in addition, of course, highlighting, say, point number 3, the payload ratio is very easy controlled because these are chemically produced. That also contributes to a simpler manufacturing and high purity. Because of their small size, the potential for higher tumor penetration certainly exists. And then there's also potential for less negative effect on these target receptors, functions, expressions, and trafficking compared to utilizing some of the other antibody-based platforms to deliver these kind of compounds. And one of the larger advantages is probably around the renal clearance versus antibodies actually staying in the body for a long period of time and being cleared either by the liver or spleen. So we are actively expanding our pipeline, both partnered programs and in-house for highly selective cancer targeting peptides for use in these radiotherapeutics and radiodiagnostics. So if you go to the next. So this is just highlighting a little bit some of the current partnerships and advancement of programs. We have 2 main partners thus far in the RI-PDC space. Reid Bio, which covers a number of programs. And here recently in December of 2022, we just announced the first election or nomination of the development candidate, which is now considering progressing into humans and designing and considering those trial designs. And an important note for the Reid Bio programs is also in 2022, we regained the rights, the Japan commercialization rights to all of the Reid Bio partner programs. So as these programs advance into the clinic in the US, we will retain Japan rights and look forward to developing those through PDR Pharma in Japan. And we're happy to talk more about that maybe in the Q&A session. The other large partner is, of course, around Novartis, where we have multiple programs ongoing. This is also making exciting progress and success across a number of these programs. And we are looking forward to announcing the first development candidate sometime here in 2023 as we head into the new year. So we think this will be the first of a number of programs to actually advance into the clinic from both of these partnerships. In addition to this, of course, we are working on our own internal RI-PDC programs. And we are hoping to probably announce the first development candidate in 2023 for one of our internal programs. The goal of that program, of course, is we will maintain or retain Japan rights, Japan commercialization rights, and likely license out ex-Japan rights to another company, on which there are a number of interested parties at current. Once we have more information, once we've decided the development candidate, we will actually announce the target and indication and provide more details around each specific program. But we have a number of programs ongoing that we're kind of looking to prioritize and determine which one can advance the fastest. And so in addition to that, so we have 2 main partnerships around Reid and Novartis, as I mentioned here, but we're also in discussions with a number of other companies who are very much interested to get into the peptide RI conjugate space. And so there will likely be more news on this coming in 2023. And so this is just to show some of the illustrated data that we've seen that I think is very compelling, and in part why we're so excited about the being able to use peptides to deliver RI drugs. And as you can see, we have a depiction here of actually giving this to an animal mouse model, a xenograft mouse model, to which you have an orthotopic implanted tumor, which is highlighted here. And you can actually see the utilization of the peptide delivers very effectively the RI compound to the tumor, specifically to the tumor, and really not going anywhere else. Of course, as also high at the bottom, you can always see kidney because these are renally cleared, and so you'll see some level, of course, of the signal showing up in kidney as the compounds are being cleared from the body. And when we translate this to efficacy on the lower panel here, when we monitor the tumor volume of these animals, you can actually see the tumor volume drastically decrease. In comparison to the red line, of course, is the non-treated animals, to which the tumor volume basically increases 3 to 4-fold over the 21-day study. By dosing the RI-PDC conjugate, you can see the tumor actually shrinking to about 1/4 to 1/5, almost completely going away upon the treatment of these compounds. And so this is a very, very exciting space. Again, there's already high validation for using macrocyclic peptides, as Lutathera is a macrocyclic peptide targeted radioisotope drug. And so we're very excited to see how these programs will progress and move forward to the clinic. A second major payload area for PDC programs, as I mentioned, is around using oligonucleotide drugs or oligo drugs. And so on the left is just kind of highlighting some of the past history of oligo drugs. In the 2000s, many companies were actually the power of siRNA or oligo therapeutics to actually knock down a target gene and suppress the production of that target gene's disease protein has been known for quite some time. This is more than 20 years now. What was very difficult was actually to translate that into a viable therapeutic that you could use actually in humans. In part because the siRNA doesn't have any real targeting function. They're not very stable in the body. They'd quickly be degraded or cleared, making them not very useful as a therapeutic. And so people really looked at a lot of LNP or liposome kind of formulation technologies to try to deliver these siRNAs into, say, targeted cells and or tissues. Even that was quite challenging. These LNP particles in liposomes, certainly this is 20 years ago, were not as good as they are nowadays. They certainly continue to improve over and over or every year. But back then, there was a lot of manufacturing challenges. They're not very stable, and they would often fuse together, so making it quite difficult. In the 2010s, Alnylam, which is certainly the leader, I would say, or one of the leaders in the siRNA space, developed the GalNAc technology. And what they did was they actually attached these sugars to the oligonucleotide drug, and there's receptors in the liver that actually grab these sugars specifically selectively and actually internalize. And so by attaching these GalNAc molecules to their siRNA compounds, they were actually able to target the drugs to the liver, effectively target them to the liver. And that has led to the development of a number of novel liver disease treatments, and also the dramatic pipeline expansion of their therapeutic compounds. And so as now we head into the 2020s, the field is pretty good at treating liver disease, but is looking to expand beyond liver disease, right? And so it's how can we start targeting, say, kidneys, lung, heart, muscle, and brain are probably the 5 other main organ spaces that are of the highest therapeutic interest at this time. And so to do that is using macrocyclic peptides. It's turned out that macrocyclic peptides seem to be quite powerful when we conjugate on an oligonucleotide drug or an SIR drug that we can use these macrocyclic peptides to target to specific cell populations and or tissue types, see internalization and then specific delivery of the oligo drug, and actually target gene knockdown as a readout. And so the power of this is that you could actually find then, so just as GalNAc is being used, conjugated to these compounds to send them to the liver, that we could find peptides that can target these siRNAs to these other organs of interest, such as kidney, brain, lung, heart, and muscle. And this would really allow you to do the highly selective targeted delivery and allow for the expansion of a number of these siRNA therapeutics. And so that's really kind of a powerful aspect to this. And so if you go to the next slide, so we have partnerships already with Takeda and Alnylam in this space. We had initially shown that our TFR binding peptides were quite effective at delivering an oligonucleotide drug to the brain and also to muscle. And actually, we're expecting the first development candidate to be announced here sometime in 2023, originating from our Takeda relationship. In addition, there's multiple programs in the neuromuscular and CNS space currently progressing, utilizing these TFR binding peptides. And again, we've announced the 2 deals for both neuromuscular targeting and CNS targeting with Takeda in prior years, 2021 and 2022. With Alnylam, we have multiple programs ongoing, looking for, again, peptides that are capable to deliver to non-liver tissues and or organs that are currently progressing. And so we're very excited with the progress we're making with Alnylam on this finding alternative peptide carriers. In addition to that, we've started up some internal programs, also looking to be proactive here, as there's growing interest in using our macrocyclic peptides to deliver these oligo drugs. And so we have identified a number of interesting carrier peptides, potentially for use in delivery into the CNS, across the BBB, that we hope to actively build a business strategy around. And so, as I mentioned, we are anticipating the first kind of development candidate nomination with Takeda in 2023. And then we're also in discussions with a number of new oligo PDC partners, not only for our TFR peptide carriers, but also other carriers that are currently under development. So I think everyone should expect additional news on these aspects in 2023. And this is just to highlight some of the data that we've actually seen using these peptide carriers. So on the left side here is when we utilize our peptide, our TFR conjugated peptide, we conjugate this to an siRNA. We can actually see a very nice shift in cell binding. So seeing the red bar move means that this specifically binds to the TFR expressing cells. And on the right side is the TFR minus, represents a cell line that doesn't express TFR. And you can see that our peptides clearly bind and carry this siRNA to the cell type that expresses TFR and not to cell types that don't express TFR, showing its kind of selectivity. This is further highlighted with a different target. Target X is just an undisclosed target that our peptides are binding to and able to deliver this siRNA specifically to upon having the target of the peptide. And you can see, so if the siRNA, which is covered in blue here, does not bind to the cell, when we conjugate our peptide to it, now the peptide actually delivers the siRNA specifically to the cell. And so when you can see on the right side, which is a negative cell line that doesn't express this target X, you can see that there's no targeted delivery happening whatsoever, and both of these molecules simply don't go in, regardless of whether the peptide is attached or not. So this actually shows a very highly selective ability to use the peptide to deliver the siRNA specifically to the target cell of interest. And then this translates on the right side to actually effective knockdown of the target gene. So MOC in this case is simply just the siRNA or the oligo drug only, and the oligo drugs will not classically cross the cell barrier of the cell membrane and enter the cells. And so if you just give siRNA by itself to these cells, you don't see any gene knockdown whatsoever. If we conjugate those siRNAs to a control peptide, so one that is not selected for a target of interest, you also see no knockdown. But when we actually conjugate the siRNA to cell target specific macrocyclic peptide, in this case TFR on the upper side, and to target X on the lower side, you actually can see selective delivery of the siRNA drug and effective gene knockdown, target gene knockdown, which is very critical. So this is really kind of a nice ability to utilize these peptides for targeted delivery and delivering oligo payloads, a wide range of oligo payloads, again, to various target cells, both in vitro, and we also have in vivo activity, of course, showing this same attributes. In the next section is an area that we're also moving into, which is, this is more similar to everyone's information to the ADC space, of course, more classic antibody drug conjugate space, is the utilizing peptide replacements for those antibodies, or just utilizing peptides instead to deliver a cytotoxic payload. Of course, as just shown on the left is a rough history of the, say, targeted delivery of cytotoxic drugs. There's a number of cytotoxic small molecule drugs on the market, but they had very poor, in many cases, have very challenging side effect profiles. They've slowly gotten better. So chemotherapy, of course, is a standard small molecule drugs that, of course, have cytotoxic inducing effects. But what you see is often you see those small molecule compounds going everywhere in the body because they're not specifically targeted just to the cancer cells of interest. They're actually also going into normal cells across the body. And so the goal, of course, for these cytotoxic molecules for the treatment of cancer has always been to more selectively to target or selectively deliver those compounds more specifically to the cancer cells only and not to the normal cells in your body. And this kind of led to the advent, of course, of ADCs because antibodies were well known to be quite selective and specific for targeting certain cell types. Companies started to look to conjugate cytotoxic drugs onto these antibodies. And Kadcyla in the HER2 space by Genentech was one of the first in this area, which is almost 20 years ago now. And what has then improved, so there were still challenges even back then. They had challenges around actually the payload potency. They had challenges around antibody-drug antibodies, or ADAs, which you'd see. The linkers weren't quite as stable, and they had some challenges with of course, just labeling these peptides, these antibodies to begin with. You've seen slow advances over the last 10, 15 years across this space. Everyone's probably aware of the Daiichi Sankyo, which really took the HER2 base antibody that was in the original Kadcyla, and now in Enhertu, this is a much more efficacious compound. And really, they worked on a much more stable and cleavable linkage strategy, and also changed the type of cytotoxic agent that was being used in those molecules to make that a much better therapeutic compound. Regardless of that, there's a number of good examples with ADCs working quite well, but there's only about 10 or 11 approved ADCs on the market here in 2022, and there's still a number of viable cancer targets that are probably going to be difficult to treat or target with an antibody-drug conjugate. And so we actually believe there's a number of companies, of course, out there looking for other modalities besides just the antibody to actually specifically deliver cytotoxic agents, of course, to potentially both improve efficacy and also safety. In addition to that, I think to open up additional target space for this modality. And so certainly using macrocyclic peptides is one large opportunity, we believe. We know this already from our RI payload space and from our oligo payload activities, that the peptides are very good at targeting specifically to the tumors. And therefore, whether we deliver an RI drug or an siRNA drug or now a cytotoxic payload drug, the concepts are actually quite similar in what you're actually trying to do, that you'd see the peptide delivered, bind to a specific target expressed on the cancer cell, that would then be taken into the cancer cell and deliver specifically the cytotoxic payload. Some of the advantages, of course, of this, these are similar to the RI and oligo payload benefits, but because macrocyclic peptides can be, of course, the properties can be highly tuned, you can improve on these compounds quite quickly. You can also manage kind of the labeling ratios, whether you have just one cytotoxic bound or multiple cytotoxics conjugated to your peptide is something that can be precisely and easily controlled. Again, the opportunity here for higher tumor penetration, or it's certainly different distribution kinetics, and then the ease of manufacturing and quality control. And of course, the last benefit is there's certainly greater IP space, as there's a number of companies working in the ADC or ADC-like conjugate space. And so we feel, of course, we're very, very confident in utilizing these peptide carriers to have the potential to both improve efficacy and safety, and actually increase the spectrum of available targets that can be used in this space. And so we will be announcing, we're in discussions with a number of new partners to move into the cytotoxic PDC space, which everyone should be looking forward to here in the coming months and into 2023. The last area I'm going to talk on is kind of a newer area for us. Well, in a way, it's been a couple of years, but is the multifunctional, what we're now breaking apart into the multifunctional peptide conjugate space. And we're doing this in part because to us, the peptide drug conjugate space is actually using the macrocyclic peptide to deliver specifically a drug payload. In the multifunctional peptide conjugate space, you're really using these peptides to be bifunctional or trifunctional or bridge things together. And so the Biohaven drug that used to be previously with Clio, and then of course as Biohaven bought Clio, is our CD38 targeting multiple myeloma drug, is in fact one of these bifunctional molecules, which we'll touch on briefly here. So go to the next slide. This is kind of just outlining some of the concepts of multifunctional peptide conjugates, or MPCs, I'll call them. Everyone might be aware, of course, there's a growing trend toward moving toward bispecific antibodies, of which an antibody, actually one side of it can bind to one target, shown on this slide, conceptually shown on this slide as orange, and then the other side of the antibody can bind to another target. And this has been a quite exciting space, and there's already a few approved bispecific antibodies that have now reached the market. Amgen, one of the largest initial players in this space. And you've seen this evolve from just the full bispecific antibody to actually into what are kind of listed here as next generation antibodies. So these are now taking kind of the Fab portions or the scFv portions of a normal antibody, and then linking them together in various configurations to make bifunctional molecules. So the BITE is shown here is quite a famous representation out of Amgen of actually connecting these two of the end parts of the antibody together to make a bifunctional molecule. There's also now evolved into the trike space, which is actually a trifunctional molecule. And then, of course, we've listed DART, TriTacs, di bodies, and tandem antibodies. So there's been a number of these kind of evolutions that are being investigated by various companies and are moving into the clinic. And the general concept is that you're making something that is either bifunctional or trifunctional to have represent a new modality. And so shown on the right is actually macrocyclic peptides. Our macrocyclic peptides are almost ideal to do the same type of concept. As we've stated many times, the macrocyclic peptides have the same selectivity and affinity. You can make them to have the same selectivity and affinity as antibodies. And therefore, it's also, because they're chemically bound, easy to link 2 peptides together, link 3 peptides together. Even now, we have linker technology to link 4 peptides together and really kind of make these bispecific, trispecific, or multispecific kind of configurations and combinations. And so we've kind of listed on the slide on the right, you can do 1-to-1, you could do 1-to-2 ratios, 2-to-2 ratios, 3-to-1 ratios. There's a lot of different ratios that could be done to connect these macrocyclic peptides together to make some very compelling new multifunctional modalities. And so you're really just kind of flipping out the scFv or the Fab portions of these other kind of concepts, replacing that with a macrocyclic peptide, which has significant benefits, of course, is our ability just to make these compounds, find these compounds. And so the second bullet point there is a quite powerful one. So because we actually can find these macrocyclic peptides to the various targets individually first, and then after conjugate through the linkers, this is actually a much more rapid way or a faster way to make these type of molecules in comparison to, say, the next generation antibody or ways that those are discovered. And so this should be quite powerful for us to actually see. And it's just that, again, because they're chemically synthesized, you don't have the challenges of the platforms shown on the left, which are all biological in their basis. So we believe there's considerable ability, of course, for faster development times at lower costs. And of course, there's greater freedom to operate, of course, in the space because there's a number of players on the space in the left now. And so we possess, it's also interesting because we possess a number or a growing number of high affinity selective peptide binders to a wide array of relevant therapeutic targets, which we can now leverage to create these bifunctional, trifunctional, multifunctional MPCs. So this is extremely modular for us and flexible as a platform. And of course, because of our large pharma connections, we've discussed these ideas with the big pharma partners, and there's considerable interest in what we can do in this space. And so on the next slide is just one example, of course, is the program with Biohaven, the CD3, is actually, of course, two peptides linked together. So this is a bifunctional molecule on one side, which is not represented the prettiest in the depiction, but on one side of this is a antibody recruiting molecule. It's a macrocyclic peptide that binds to circulating IgG. And then on the other side is a macrocyclic peptide that actually binds to CD38 on the surface of multiple myeloma cells. And what this does is it actually allows for the targeted recruitment of basically NK cells to the cancer by way of grabbing the FC portion of circulating antibodies. So what they do is when they treat these patients, what's happening in the clinical trial study is actually you give the patient, you treat the patient, with exogenous NK cells incubated with an IgG antibody and our molecule, our bifunctional molecule. And you can see in the middle depiction here as this BHV110. What that does is the antibody will actually bind, the FC portion of the antibody binds to the surface of these NK cells. Our molecule binds to that other side of the FC antibody. And then when you then deliver these to patients, our bifunctional molecule allows for the recruitment or carrying of those NK cells directly to the multiple myeloma cells in circulation. And you see considerably increased lethality upon doing this compared to just giving the patient, say, NK cells by themselves, which don't have any kind of specific targeting function toward the multiple myeloma cells. And so this is actually, of course, at current, it received orphan drug designation from the FDA. This is back in 2020. It started in the phase 1A, 1B, which is not in healthy volunteers. This is actually in multiple myeloma patients in October of 2021. And we're expecting to be able to report data on this, hopefully sometime in 2023, which is very, very exciting. So we have high confidence in that this molecule is going to show very effective results in these patients. And so, because of course that's already a bispecific, and basically in a way that's acting to recruit natural killer cells to the cancer cell, but it's using basically an antibody as a middle portion. That this can actually be done more directly by just having one peptide that either binds to T cells or NK cells, natural killer cells, and then the other portion of the peptide actually binding to tumor cells. So it's shown in this depiction is having, again, a macrocyclic peptide that would bind in this case to HER2, which is overexpressed on certain types of cancers. And then on the other side, you have a macrocyclic peptide that specifically binds to T cells or to NK cells. You would then recruit those T cells, killer T cells or killer NK cells to then specifically attack the tumor. And this concept, again, is not new to us. The concept of T cell engagers is what they're called nowadays. T cell engagers or NK cell engagers is actually a very exciting and growing area. This is also CAR-T is kind of in this space also. And there's already known antibodies, both, again, as I just touched on, bispecific antibodies and other next generation like antibodies that are doing the same type of bridging and or recruitment type of function. And so for us, we already consider significant benefits of using the macrocyclic peptide versus using antibody or next generation antibody platforms to do this. And it's shown on the right side of this is actually, again, replacing the pieces and parts of this type of conceptual molecules by actually making these bispecific or trispecific or multifunctional peptide conjugates. And so at current, we're actually increasing the number of T-cell specific binders and NK specific binders that we have. We already actually have a large pipeline of tumor targeting peptides. Again, many of those are coming from the RI peptide drug conjugate or the cytotoxic drug conjugate space. And those peptides could be utilized also in a targeting capacity to recruit both T-cell and NK cell. So instead of, for example, bringing a payload that's attempting to just kill the tumor cell, you're actually recruiting your own immune system to come and kill the cancer cells. And so this is an extremely powerful area for us, we think, going forward, and is an area of focus in 2023 in this kind of MPC space, and something we look forward to disclosing and talking about more. The last couple of sections is the PDR Radio Pharma business. So as everyone might be aware, the business is doing extremely well. The team here has done a fantastic job in already turning that company, which was operating marginally in the red, to operating now marginally in the black, but it has a significant improvement. And there's a large focus on getting PDR Pharma to become a significant revenue contributor to PeptiDream. The company already currently sells 8 approved radiotherapeutics and 24 approved radiodiagnostic agents in Japan. And we are working hard to promote both the use of those compounds and also indication expansion of the approved products here in Japan. In addition to that, the pipeline itself, there's actually 4 different programs in the PDR Radio Pharma pipeline. Recently, they have just announced a deal with Eli Lilly to bring in Talvid, which is a PET imaging diagnostic for Alzheimer's disease. And PDR Pharma will be co-developing that in Japan with Eli Lilly. And of course, Eli Lilly has all the ex-Japan rights, hence this is listed in gray. And this has already been approved outside of Japan. And the work is to, of course, get this approved in Japan for being able to diagnose Alzheimer's disease in patients. In addition to that, we also have a PSMA program that is currently ongoing that is actually in phase 2 here, that was in-licensed from Lantheus Medical. So Lantheus is in a phase 3 in the US with this compound, and to bring this compound into the Japan market had to go through PDR Radio Pharma, which is supporting the efforts there to develop this drug also in Japan. And then there's 2 additional compounds around an integrin, which is called FF10158, which is actually a therapeutic for malignant gliomas and potentially some other cancers. This was actually out-licensed to Novartis, which is actually running this in a phase 1 at current in the United States and in Europe. And we retain the Japan rights for this compound, but it hasn't entered the clinic yet in Japan. So what classically will happen is this is developed outside of Japan, we'll actually use bridging studies to bring that compound into the Japan market, which we can do with less patients at a lower cost and then accelerate its development. And then there's one additional therapeutic, which is a cadherin-3 targeting agent that is currently in a phase 1 study, both in Japan and in the United States, and is something that PDR Pharma co-owns with Percells Proteomics. So these are the 4 current programs that are in clinical development across PDR Radio Pharma. And PDR Radio Pharma is expanding or focused on expanding the pipeline and product offerings basically by 3 things, of course, is in-licensing third-party products for sale in Japan. So we're in discussions with a number of interested companies who want to bring their radiotherapeutic and radiodiagnostic agents into the Japan market, which is quite a closed market, and you have to go through one of the 2 players here. that have the licenses in the infrastructure to handle these type of agents. The other is actually utilizing service agreements to distribute these type of third-party compounds for sale in Japan, where they access our infrastructure, our regulatory abilities, development abilities, for fee, of course, to bring their products into the Japan market. And then, of course, feeding our own both internal and partnered RI-PDC programs that we're developing outside of Japan to actually bring those into the Japan market, utilizing PDR Radio Pharma to both develop and then sell those compounds, such as our in-house RI-PDC programs or our Reid's partnered RI-PDC programs, to which we have the Japan rights too. And so really the goal is, of course, becoming the leader in radiopharmaceuticals and the partner of choice here in Japan. We're pretty much already established, PeptiDream is already established as a powerful peptide discovery company for RI peptide conjugates globally. And so being able to get the commercial success and kind of capture more of the financial upside of these compounds in Japan is a key consideration, and in part why we acquired the PDR Radio Pharma business. So just highlighting here is the tau, which is a diagnostic agent. It shows actually enables the visualization of amyloid beta accumulation and or tau, which are both useful diagnostic treatments for Alzheimer's disease. We already had the Amyvid agent, which is more for the diagnostic imaging of A beta accumulation. And now Tauvid is the new diagnostic agent out of Lilly for tau. And actually both Amyvid is also from Eli Lilly, and now Tauvid is, these are the two kind of critical, is believed to be the critical diagnostic agents for really being able to assess Alzheimer's disease in a broad patient population that you could actually test them with both. And so we will largely be probably discussing more about this Tauvid program and product as this advances. But the deal was signed earlier in the year here for us to, of course, co-develop this PET imaging agent here in Japan. And PDR Pharma will be responsible for obtaining market authorization also in Japan, and then utilize both of these diagnostic tests for the diagnosis treatment of Alzheimer's disease. The last topic I'll just briefly touch on is we have a number of kind of sister companies under the PeptiDream PDR Radio Pharma umbrella. One of these is Pepti Growth, which was founded in 2022 as a joint venture with Mitsubishi Corporation. Please visit the website at peptigrowth.com if you'd like to learn information. It's now online and it looks quite nice. And this company is focused on replacing proteinaceous or protein-based growth factors with peptide alternatives. And there's really a large market need to realize the expansion of both cell therapy and regenerative medicine therapeutics by switching over from proteinaceous growth factors to another replacement. And so the nice part of this business is PeptiDream also owns the therapeutic rights to the Pepti Growth products that are being developed. And so on the next slide, slide 32, is just kind of the current pipeline of Pepti Growth. And thus far, there's been 6 products launched through December 2022. Earlier this month, we just announced product 5 and product 6 on December 8th. And these are increasing sales. Mitsubishi handles the global marketing, promotion, and sales of these compounds, and PeptiDream handles the discovery and validation work of these compounds. And we have a number of other compounds that we're looking to launch, probably at least 4 more products in fiscal year 2023. And as we replace more and more of these proteinaceous growth factors with peptide replacements, we are going to see, hopefully, a dramatic increase in the revenue that these products can actually produce through the Pepti Growth sister company. In addition to that, again, as we've mentioned, PeptiDream retains the therapeutic rights to these compounds. And the first compound, our PG001, which is an HGF peptide alternative, we licensed to Genentech for therapeutic use earlier this year. And so we're actually currently under discussions in regards to the other products, Pepti Growth 02 through 06, and looking at interested parties for the therapeutic use of those candidates and peptides also. So we believe this is quite a successful relationship to expand both the regenerative medicine space, but also that we retain the therapeutic use of these compounds, allowing for PeptiDream to grow certain aspects of this business also for the PeptiDream side. So a very mutual beneficial relationship we believe we have here. And looking forward to again in 2023 launching additional products, and also potentially making updates on the out-licensing of therapeutic rights to the already launched products. And so it's just shown on slide 33 here some of the data actually that had come out of the first four the most recent four compounds that had been launched, PG003 through PG006. PG003 was a brain-derived neurotrophic factor peptide alternative. It has the same function as the BDNF protein equivalent, but now it's just a very small macrocyclic peptide with that can be chemically synthesized and purified to high yield. And so potentially its therapeutic use, because it's a TrkB agonist, it might have certain uses or seems to have certain uses in enhancing brain activity or contributing to neuronal health. And so we think it could have a role in the treatment of neurological disease, and these type of aspects are being investigated. The PG004 was a noggin, basically alternative replacement, which is a bone morphogenetic protein family antagonist. And so potential uses of this compound for therapeutics or therapeutic use is basically a variety of bone diseases and also potentially Huntington's disease. So these are also being looked at. And then PG005, which is a selective BMP7 inhibitor, has higher selectivity than noggin, so maybe potentially similar space. And also the selective PG006, which was a selective BMP4 inhibitor, potentially also has use in this space. And these are kind of meant to be listed here in the lower right side depiction about what these potentially could be used from a therapeutic setting. And so, again, very excited about these products being used by PeptiGrowth to expand the cell therapy regenerative medicine space, which are being used ex vivo, so they're being used outside of humans, of course, and then also for PeptiDream to potentially partner these programs with other partners to develop them as therapeutics for use in humans. So the last slide that we have in our deck today is just kind of touching on some of the short-term catalysts we're looking for here in the near future. In the therapeutic peptide and small molecule space, of course, we're anticipating phase 1 results from the BMS study, hopefully in 2023. Once we know more, of course, we will be very excitingly announcing whatever results we get. We're expecting the AZP 3813 GHR antagonist program to actually move into the clinic, again, probably around summer of 2023. Amylite is saying it's in the first half. And again, it's in IND enabling studies, and everything looks very, very on track for this program. PA001 with Peptide, we'll be looking to announce development plans or out licensing strategy for this program sometime in the near future. And then we have a very exciting myostatin inhibitor, which we didn't discuss today. That is a fully owned in-house program that is now advancing toward, say, the selection of a development candidate. And we're also planning for next steps in regards to that program, which we will be announcing once it's decided. And then, of course, the modules program, which we're expecting an out-licensing or partnership deal sometime in early 2023 to be able to be announced. On the therapeutic PDC side, we've just recently announced, of course, the first development candidate in one of the programs we have with Reid Bio, and looking forward to that to move forward and hopefully into the clinic at some point here. And then across Novartis, we have multiple programs with Novartis also, but actually the first program has advanced to selection of development candidate here. And so as we reach that milestone, we intend to, of course, announce that as soon as it's available. And also announcing a little bit more information on the first in-house programs for RIPDCs is hopefully arriving at our election of our own first development candidate for us to be considering for development. Across the oligo PDC space, I think one of the more earliest catalysts will be around the Takeda, and those programs are progressing extremely well. In the neuromuscular space, we should have a development candidate nomination here sometime in 2023. So we'll be excited to announce that as that progresses toward moving into the clinic. And then on the therapeutic MCP side, again, the current most advanced program is the program partnered with Biohaven for the CD38 multiple myeloma drug, and hopefully being able to announce on the phase 1A, phase 1B results. Again, these are in multiple myeloma patients, so eagerly anticipating the ability to release these results when they're available. We have a number of program partnering, out licensing, and new deals in various stages of discussion. The interest, of course, across both the peptide therapeutic and the kind of peptide drug conjugate and MPC space is as high as it possibly could be. It gets higher every year, it seems. We have a number of companies interested in RI conjugates, in oligo conjugates, and cytotoxic peptide drug conjugates, in addition to doing new peptide therapeutic programs. And so we think it's, again, the business is in the best condition it's ever been, certainly, and we're looking forward to a very exciting and productive 2023. On the last section of PDR Radio Pharma, there'll be new additional announcements coming out of PDR Radio Pharma as we're looking at a variety of in-licensing deals, service agreement type deals, and also advancing the current development pipeline, and of course, feeding products that come out of our relationships into the PDR Radio Pharma pipeline. Should be exciting announcements here coming in 2023. So